John Christie, and he had this notion that the Defense Department would be interested in where people would choose to put something in a blank space. He had fairly worked out ideas about this, and wrote a proposal. I had to help him put it forward to the Defense Department, because the Defense Department doesn't give graduate students research money. Actually, they were quite intrigued. You can sort of think of this a little bit like the game of Battleship. So you've placed a battleship on the board, and I only see the blank board, and so I'm now going to guess where your battleship is, and I'm going to try to find it with a series of guesses. And the question was, when you ask someone to place something in a space, like could be a square or a circle, we had different shapes that different shaped fields, randomly, do they behave randomly, or is there some consistency in their responses? And it turns out that the shape in which you're locating things has a very significant control over the choices that people make when you ask them to behave randomly. And not only did we ask people to place things randomly in these spaces, but we also asked them to place them where they thought most people would place them. And we also asked them to place them where they thought most people wouldn't find them. So we think of it as a hiding task, can you represent what's in most other people's minds and choices task. And I think part of what we found was that you could actually get a reasonably good representation of where most people will place things by asking people where they think most people will place things. The shape of the field matters. Perhaps that's sort of obvious. If you ask people to place things in a square, there's a tendency for things to go along the edges and across an X shape. In a circle, it also goes all around the edges, but then there's no X shape anymore, and so and then the middle tends to get some extra choices.
So why was the Department of Defense interested in?
That's a very good question. We told them they should be interested because it might help them figure out where to put a base in the desert or hide something in the ocean. But only the Defense Department knows why they were really interested.
And then we also saw that more of the recent stuff is about this inhibition of return. Do you want to explain?
Yeah, we might say that half of what the lab does is related to inhibition of return. If you imagine that you're searching for something, and it's the kind of search where you sort of have to look from place to place. Like, let's say I asked you to look around the lab and find a picture where it looks like Star Trek, but actually it's members of the lab. You may or may not have seen that one. Mm-hmm. So you look at a picture and say, well, that's not a Star Trek picture, and look at another one. And eventually you find a Star Trek picture, but it's not members of the lab, and you'd keep looking. So one of the questions is, how do you stop yourself from returning to where you've been before? And, well, one method you could do is you might actually have a very rigid strategy for your search. Like, you might say, well, I'm going to do the upper left to the lower right, and I'm going to go down like this. But if you didn't have a rigid strategy, and most searches are like that, it would be good if you had a mechanism that would help you remember where you'd been before and discourage you from going back there. And inhibition of return was a phenomenon that was discovered in the mid-'80s by Mike Posner, actually, and his grad student, Yoav Cohn. And what they found was that in a queuing paradigm, which is where there's a flash of light that actually doesn't tell you anything about what's going to come on the screen next, and then there's a target and you have to process the target, you process the target faster if it's where the flash was, if it occurs very shortly after the flash. But if it occurs after a delay, like maybe after half a second, you're actually slower where the flash was than in any other location in the field, equivalent location. So the original attraction of attention to that location was called attentional capture, and it produces facilitation of performance, but after you wait a while you see this crossover and the performance becomes worse at that location. They named that inhibition of return and described it as a mechanism that was probably good for enhancing our orienting to novelty. And studying that a bit, I thought, well, that might be the mechanism that helps our search be efficient. And so I've been quite interested in that idea and have done quite a few studies exploring it. For a period of time I thought the idea was wrong, so I did a lot of studies suggesting it was wrong, but subsequently we're convinced it's probably right. Exactly how inhibition of return works and accomplishes this function is debatable. And so there's, let's say, room for research there. I've collaborated with people who do a single unit recording in the monkey to look at the neural mechanisms of inhibition of return. And I'm collaborating with other people now to look at using ERPs, basically recording brain waves, to see if we can get data from people on the mechanisms. And that would tell us a little bit about which brain structures are involved in laying down these traces or storing them. It might not be the same brain structures. Work with patients can also be used for informing us about this. And I haven't actually done any work with patients and inhibition of return, but some of my colleagues have.
Okay. Is there anything else you want to tell us about your research before we do the tour of the lab?
No, probably not.
Okay, so
My wife and I, as I said, came to Halifax in '74, but we didn't think we were ready for kids right away, so we actually didn't have kids, and we had been married since '70. We had our first child. We actually conceived our first child on sabbatical. That was in 1980, and Sarah was born in 1981. And so she's 26 now. 2 years later, we had Lauren. At the time we had both our girls, we had an Afghan hound, so our family was, you know, I don't know what they call it, you know, 2 adults, 2 kids, a dog, and a car.
the typical family?
Yeah, maybe that's a typical North American family. And since then, we've had 2 bearded collies. We have our second bearded collie now, and the girls grew up and went off to college. One is in music and still doing music actually at the master's level at UVic, the younger one, and the older one did cultural studies at Trent, took a year off to teach in Japan, got her master's degree from NYU in a field called performance studies, and now is kind of doing both performance and TAing in Peterborough and Toronto. And in fact, we just heard the other day that her theater group that she's working with was invited to a puppetry workshop in Turkey.
Oh, wow.
And she's going to be going there in May, and she may be coming here to Halifax to do some musical thing with a musician where she does something visual while the musician does something auditory. So that's pretty exciting. I'm married to Marilyn. We got married in 1970. There's a whole other story there. And Marilyn works in the Faculty of Dentistry. She's the audiovisual department. She was originally hired to be their clinical photographer, but as the field of photography evolved and as their division of instructional resources evolved, she became very involved in all kinds of other things, including maintaining their webpage and helping the dental faculty generate posters and materials for teaching. And so she's pretty much a multimodal audiovisual person now who still does some photography.
So what do you do in your spare time now that the girls have left?
Okay, so I am in the very fortunate position of having research be my hobby. So, because I spend most of my time doing research, I'm enjoying it 24 hours a day. I do, of course, and I don't think we mentioned this, I do teach. I teach at the graduate level, I teach at the undergraduate level. And in fact, next year we're starting an undergraduate class on attention, so I'll be teaching that.
What about cooking? I heard you like to cook.
I love to cook. I also play bridge. Maybe those are my 2 hobbies aside from research. I don't play that much bridge. Maybe once or twice a month I play bridge. And in fact, the last trip my wife and I took to Toronto, we went to see Sarah's show, which was called What Alice Found There. It's about Lewis Carroll. We did do a cooking class at the Calphalon Culinary Institute, which was tremendous fun, and then we came back here and we made all those dishes for our friends.
And you seem to be a Star Trek fan, so...
Yeah, when I was in college, Star Trek was on, the first Star Trek, and I mean, I vividly remember getting together in somebody's living room to watch it every Saturday. And I would say we've been somewhat addicts ever since, although I think we've become successively disappointed as the shows have, like this, the older ones are still better than the new ones. I think we like New Generation, and the latest one, I can't remember what it's called even, Enterprise, it was not very good. So, but yes, we're fans, and that's just because it was a groundbreaking show. partly because of the science fiction and partly because of the humanistic and cultural themes. So in a lot of the research we do in my lab, it's important for us to monitor where people are looking. Sometimes we don't want them to look. We want to study attention without moving your eyes, and so we actually tell them, don't move your eyes, and then we can use equipment to tell them they have moved their eyes, and then they'll learn not to do it. But this equipment, which can record eye movement position really accurately, and Patty will help put this on, is called the iLink 2. And one of my colleagues at University of Toronto invented this, and he markets it. It turns out now that one of our former honor students is his chief executive officer of that company. And so when we have any questions, I call up my former honor student and get the answers. So Patty's going to hook this up so that she can basically use the apparatus to monitor where I'm looking, and then we'll illustrate a very simple experiment that we've done. So the first thing is, we try to image my eye on the screen. So when we have a reasonable image, what the software in the computer does is it's making this video of my eye. It actually can do it at 250 hertz or every 2 milliseconds. And it figures out where my pupil is by looking for basically the darkest image. And so it can figure out where my pupil is, and you can see as I move my eye, my pupil is turning. It can figure out where my pupil is in relation to what angle it's pointing. There's a infrared light source here, and there's photocells here that pick up the infrared light. That allows the computer's software to figure out where my head is in relation to the screen, and by using both those pieces of information, it can figure out where I'm looking on the screen. So that's a 9-point calibration, and we do it again, and the computer uses the values that it's getting, where it thinks I'm looking, to see how good a calibration it was. So this is an experiment in which we're showing our participants works of art that we collected from the NASCAD library. And in each work of art there's an interesting scene, and then there's someone looking somewhere. And we collect 7 seconds of eye movement as we tell people in that condition, look at this picture as if it were a work of art, of course, which it is. As an example, the fellow in the middle there is looking off to his right or our left, but in the original picture he was looking straight ahead. And so what we've done in this experiment that Christy masterminded is we're interested in whether the characters in something that you're looking at, which could be a real world scene, but in this case is art, whether their gaze direction affects where you look. So we took works of art in which there were individuals we considered to be, let's say, key to the meaning of the picture, and we had them either looking to the left or to the right. And you might right away think, well, you know, the artist is kind of controlling where you're going to look by how they created the picture, and there's no question that that's true. But for every picture that the artist created, we can still have 2 gazes, one towards the left and one towards the right. And if that gaze has no effect, that's telling us that the gaze of a key character doesn't really matter to where our observers look. But if the gaze has an effect, then it can't. Obviously, it's overcoming whatever is of interest in the artwork or in the scene. And so we just tested our last subject in this experiment, so we'll know the results soon. Jean-Luc befriends, or is it him when he becomes a Borg? But sorry, the fact that I even know that makes me a little nerdy, doesn't it? Hey, I'm Ray Klein. I study eye movements and attention.